Every flu season brings a new set of influenza virus strains. Carried by aerosols or lingering on contaminated surfaces, influenza viruses follow their inherent genetic programming in order to find a host and multiply within it. Fortunately, the virus carries some prominent features on its surface that are recognized by the human immune system and trigger an effective defense mechanism. Vaccines train the immune system to recognize a potential threat even faster and instantly initiate a response. Due to this increased alertness, the large-scale invasion and multiplication of viruses in the body can be intercepted at the very start of infection. The outbreak of the disease and its accompanying symptoms are prevented in most cases, or are much milder if they do occur. The production of influenza vaccines starts with the multiplication of influenza viruses in either egg or cell culture. After harvesting the viruses, the production of the vaccine can follow different paths. Historically, whole inactivated influenza viruses were used. Later, centrifugation steps were introduced to purify the vaccines, and chemicals were used to split the virus, leading to the introduction of split vaccines. In subunit vaccines, additional purification steps separate the antigens HA and NA from the remains of the virus body. Both split and subunit vaccines proved to be somewhat less reactive and immunogenic than whole virus vaccines. The formulation of the virusomal adjuvanted Inflexal V vaccine takes the development a step further. As for all influenza vaccines, the production starts with the growth and harvest of viruses. In the production of Inflexal V, the virus particles are then completely disassembled. First, they are split by adding detergent, which causes the virus envelope to dissolve into the individual components. The inner core, consisting of viral RNA, nucleo and matrix proteins, is set free and removed via centrifugation. Up until this stage, the process is comparable to the production of a subunit vaccine. In the production of Inflexal V, it is followed by the addition of phospholipid, which facilitates the formation of virusomes in the final step. When the detergent is removed, the purified elements spontaneously form virusomes, virus-like hollow vesicles. The virus antigens, surface glycoproteins HA and NA, are anchored in the virusome membrane. Once the vesicle-forming process is completed, the solution contains a homogeneous population of vesicles all around 150 nanometers in diameter. One advantage of the production process is the high degree of purity achieved. The virusome represents a lifelike but harmless replica of an influenza virus. Like the virus itself, it carries HA and NA on its surface. These antigens enable the virusome to mimic natural infection. It enters cells using the same mechanisms as an influenza virus, and therefore the immune system perceives it as a virus. Due to this structural and functional similarity to the influenza virus, the virusomal adjuvanted Inflexal V vaccine presents antigens in a natural way. Thus, the adjuvant function is embedded in the very structure of the virusome. Every year, the WHO and EMEA issue a recommendation for the influenza vaccine composition and select the 3 different virus strains to be included in influenza vaccines. Therefore, Inflexal V contains 3 different virusome types, each one of them carrying the still functional surface antigens of one of the 3 currently circulating influenza strains. In order to initiate an immune response, the virusome particles need to interact with specific cells of the immune system: antigen-presenting cells, APC, T helper cells, and B lymphocytes, B cells. The starting point of an antibody response is the activation and priming of antigen-specific B cells. Virusomes interact directly with the prototype antibodies on the surface of the B cells. Since any antigen displayed on a particle surface in a repetitive order represents a strong activation signal for antigen-specific B cells, the concentration and spatial proximity of antigens on the virusome will initiate extensive attachment and activation following a point contact with a B cell. This so-called Velcro effect also contributes to the adjuvant properties of the virusomes. B cells ingest the viral antigens recognized by their prototype antibody, process them, and subsequently display the resulting peptides using the MHC2 complex. For a full activation of B cells, a second signal is needed. At the beginning of this signaling cascade, antigen-presenting cells, APCs, interact with virusomes. The virusome's resemblance to virus particles facilitates a swift uptake by APCs. After ingestion and processing, the peptides originating from virusomal antigens are presented using MHC2 molecules. Antigen-specific T helper cells are primed by binding to the virusomal antigens. Contact with primed T helper cells triggers the full activation and differentiation of B cells into antibody-producing plasma cells. Cytokines, signaling components released by T helper cells, further stimulate multiple cell division and differentiation. The process snowballs and quickly results in a large pool of antibody-producing plasma cells. This cascade of immune events is induced by the virus-like features of the virusome. In the process of stimulating the immune system, the virusomes completely disintegrate in the human body, and only the protection remains. Due to this biocompatibility, InflexLV has an excellent safety and tolerability profile in both healthy and immunocompromised adults, the elderly and children. It is therefore suitable for all age groups.